Journal of Ecology and Environment article. Title is Occurrence of an invertase-producing strain of Aspergillus niger LP5 isolated from longan pollen and its application in longan syrup production to feed honeybees. In northern Thailand, the longan flower is the principal nectar source for honey production. Microorganisms play a critical function in the agricultural ecology. The morphological characteristics of fungal species found in longan pollen were studied. Aspergillus spp. Were found to be invertase-producing strains and were employed in the longan syrup production process. The purpose of this study was to evaluate the effects of invertase-added longan syrup on the adult honeybee population numbers that were fed by this syrup for 16 weeks. Different fungal species were found in longan pollen samples. Aspergillus was the main genus, with three predominant sections: nigri, flavi, and turai. Other isolated species were Trichoderma spp, Rhizopus spp, Neurospora spp, Cotomium spp, Fusarium spp, and Penicillium spp. However, Aspergillus spp. Is the only fungal species that produces the enzyme invertase. The invertase-producing strains belonging to the Aspergillus section nigri were found to be a niger LP5 with an optimum activity at pH 6.0 and 60 degrees Celsius. When a niger LP5 invertase was used for longan syrup processing, the highest levels of glucose and fructose were found in invertase-added longan syrup, while fresh and boiled longan syrup had lower contents of both sugars. The sucrose content was detected in at 4.25%, while in were at 4.02% and 3.08%, respectively. An appropriate amount of sugar to feed and maintain the honeybee population was considered. The data showed no statistically significant differences between the two selected forms of longan syrup compared to the sugar syrup examined by the adult honeybee population. The main species of isolated fungi from longan pollen were Aspergillus spp. The discovery of an invertase-producing strain of a niger LP5 has enabled its application for enzyme utilization in the invert sugar preparation process. The adult worker bee populations fed by longan syrup from both boiled and invertase-added sources showed an increasing trend. Artificial syrup made from longan fruit to feed honeybees when natural food sources are limited can be applied. Thank you for watching Journal of Ecology and Environment article.